The landscape of vaccine development includes different types of platforms or technologies. The ultimate goal of a vaccine is to present to the body a protein that resembles a piece of the pathogen, in this case, the spike protein of the coronavirus.
At the moment, there are 2 messenger RNA vaccines. So there's the one from Moderna, the one from Pfizer, both of them expressing the spike protein.
By RNA, we're giving a code to our own bodies to produce the spike protein. A couple use viral vectors types of vaccines, which are like Johnson & Johnson or AstraZeneca. Some of them are synthetically produced as recombinant protein technologies in the lab. And some of them are actually the live virus that it's been either attenuated or inactivated.
Novavax also has a vaccine that is getting ready for approval, and that is a protein plus adjuvant vaccine. So the protein is actually purified through chemical means, and then because the protein itself is not terribly immunogenic, the immune system gets a little bit of help with an adjuvant. And adjuvants are just molecules designed to help activate the immune system and make it work more efficiently.
I think the public needs to realize that we have had many years of prior experience with these technologies. We didn't really start from scratch. And that's really the basis of why we have been able to develop vaccines so rapidly.
Although the development of COVID-19 vaccine seems quick, Moderna has been investigating our platform for the last 10 years. So every cell that has a nucleus has DNA, and that's really the lockbox for all of our personal information. But then somehow that DNA information needs to get translated into proteins. So DNA is transcribed into messenger RNA in the nucleus, mRNA exits the nucleus, and then in the cytoplasm of the cell, ribosomes translate that messenger RNA into protein. So a messenger RNA vaccine just capitalizes on that natural process for protein synthesis. We've known probably for about 60 years that messenger RNA could potentially be a way to deliver proteins. We also knew it had adjuvant effects, meaning it can actually increase an immune response. It actually took that long to really develop the mechanism to be able to deliver the messenger RNA into the cells. And in fact, we deliver the messenger RNA with a lipid nanoparticle, which is really just a fancy way for speaking about a blob of fat that ensures that that messenger RNA isn't broken down by body's natural mechanisms. So when you get your vaccine in your deltoid muscle, the lipid nanoparticle also helps it target to the lymphatics. And once the lymphatics drain into that lymph node, that's really where there's a high concentration of antigen-presenting cells. And those are really the cells that are the target of this vaccine. We're delivering the messenger RNA directly into the cytosol, not altering the DNA, indeed. And the ribosomes then translate that delivered messenger RNA sequence to create a protein that's expressed on the surface of the cell. In the case of COVID-19 vaccine, it's the spike protein that's expressed on the surface of the cell. And then the rest of the immune response happens as it would normally happen if your cell were infected by a virus. Only in this case, there's no virus. There's just the messenger RNA. The spike proteins hang around for about 48 hours, the messenger RNA up to 72 hours. So they are rapidly degraded like any protein. The immune system actually has been designed to really develop the cells that then recognize that protein again upon reinfection or re-challenge. And that's really how the vaccine then prevents us from getting infected.
The Texas Children's Center for Vaccine Development is a joint venture between the academic HAT, which is Baylor College of Medicine, and its affiliated pediatric hospital, which is Texas Children's. As a vaccine center, our goal is to develop and bring vaccines that are safe, effective, but also affordable for everybody and anybody that needs them around the world for many diseases, including neglected tropical diseases, as well as some of these emerging pathogens. We focus primarily on recombinant protein-based technologies. for a couple of reasons. One, they've been widely used previously in many people, so they bring a safety record. But at the same time, they are affordable. We think that for a real global health vaccine to be deployed for COVID-19, you have to balance the portfolio. Some may be novel, innovative technologies, but some should be bread and butter technologies. The difference between RNA vaccine technologies and recombinant protein technologies is that with RNA, you're asking your own body to produce the protein that is presented to the immune system versus using a protein that you're directly immunizing an individual with. We started working on coronavirus vaccines since 2011. We started first with SARS and then with MERS. We immediately, of course, looked at what we had in our freezers, what we had in our knowledge, and the prototypes for vaccines for those two diseases were really the ones that leveraged us to be able to, in less than 5 months, have a prototype vaccine against the COVID-19. Our formulation of choice is an aluminum-based formulation, which is the adjuvant and the recombinant protein, plus a second immunostimulant. So there are many platforms of how you produce prototypes of vaccines. I think the key here is that all of them are targeting the same component of the coronavirus, which is the spike protein or a component of the spike protein. And when the body sees the real virus, the body thinks that they already know what that protein is and they already have an immune response ready to go. So we accelerate the ability of the body to tackle viruses such as the coronavirus. I think we're actually still pretty lucky that this virus doesn't mutate as rapidly and as rampantly as other viruses. But I think that this is also another reminder that that's the value of having so many vaccines being developed.
We don't actually know whether the vaccine will be able to protect against the variant or not. With many variants, actually, we've seen no decrease in neutralizing titers. But what we don't know is what that means in terms of the vaccine efficacy. So Moderna is undertaking an exercise to investigate what happens if we swap out the sequence for the original Wuhan strain, and those studies are actually underway now, both in a priming series and in a booster dose for people that have already received primary vaccination. Our idea is that SARS-CoV-2 vaccines could become like influenza vaccines, where good epidemiology is tracking what's happening in real time, and then we're able to put the relevant sequence into the vaccine before people would need it.
We still have a lot of questions that we do not know. So, for example, how long will the memory or the immunity last in a population? That will help us determine how often we will have to vaccinate ourselves. We may be in luck, and after the 2 doses of any of these vaccines, we don't even need a booster. I think there's still a lot of work that we need to do to understand more and more how this virus not only behaves in nature, but also how it's looking to look for its survival within us humans as hosts.